covers the renal regulation of potassium, calcium, phosphate, and magnesium, with special emphasis on potassium. Now, although we have discussed calcium in the endocrine system and GI phys, so I still expect you to remember quite a bit about calcium, but I will review some of what we did for calcium. Phosphate and magnesium are fairly easy. So we're going to spend a little bit more time on the renal regulation of potassium. Now, why is it so important to maintain these various substances? Remember, things like potassium are going to be involved in membrane potentials and generation of action potentials. Calcium's very important also in membrane potentials, in action potentials, and blood clotting, a number of different things. Magnesium, phosphate, again, phosphate's involved in, it's one of the buffers we talk about. Obviously, phosphate's needed to make ATP. Magnesium is also important in regards to certain enzymes. It's a cofactor in a number of enzymes. So it's pretty important to maintain these certain electrolytes. Now, potassium is what we're going to talk about a little bit more. Now, remember that potassium is a cation, meaning it's positively charged. It's found in heaviest concentration in the intracellular fluid. So you'll notice here the intercellular potassium concentration is much higher. And overall, when you look at potassium intake, it needs to be equal to potassium output in order to maintain balance. Most of the urine output is through the urine, which you see right here. A little bit is excreted in the feces. Now, why is it so important in maintaining the extracellular fluid volumes of potassium? Because that's really what we regulate, is the extracellular fluid levels of potassium. Well, remember, again, it's involved in establishing your resting membrane potential. It will be play a role in cell volume because of osmotic effects. It's also important for the generation of action potentials. So we have to discuss overall how we maintain the balance. We're going to look at short-term maintenance of potassium levels by looking at how we can redistribute potassium between the extracellular and intercellular fluids. Remember, potassium is a cation that cell membranes are a little bit more permeable to potassium than they are in sodium. So we can kind of move them across the membrane if needed. And this is going to be more for the short term, say after you actually eating a meal, especially if you eat a meal that's high in like fruits and vegetables, especially vegetables. Your potassium levels may be high, and we want to avoid any too high of an extracellular fluid amount of potassium. So short term, we're going to show you how we can control it short term, but long term, you got to get to the point where the kidneys. So here it says changes in intake or excretion. We can't change the intake of potassium. We don't have a potassium, appetite. We don't really like, oh, I'm going to go out and my body says I need some more potassium. I'm going to go and find some potassium. We don't do that. So mainly we control it by changing the excretion in urine. So if in your notes you can fill in these various things. This is a table from your textbook that looks at various factors that can move potassium into or out of the cells. So I want to make sure you understand the titles or the tops of the lists. On the left are various factors that can shift potassium into the cells. So it's going to move it from the extracellular fluid into the intercellular fluid. So these can ultimately, some of them, if it decreases extracellular potassium levels, if it decreased enough, you could have hypokalemia. Over on the right side, these are factors that shift potassium out of the cells. So it's going to go from intercellular fluid out into the extracellular fluid. So these things can increase the extracellular fluid concentration of potassium, which, if enough, could lead to hyperkalemia. You'll notice some of these things are going to be pathologic conditions, and some of these things would be, which specifically I want you to put a little star by insulin and aldosterone. These are going to be two factors that are going to be very important physiologically in controlling potassium in the extracellular fluid until we get to the point of getting to the kidneys and dealing with that extra potassium that we may have taken in our diet. So insulin and aldosterone post-meal will be very important in keeping potassium levels from being too high in the extracellular fluid. Again, as I mentioned before, if you had a meal that's high in vegetables, you're going to get a lot of potassium. So initially, the insulin and aldosterone are going to stimulate the movement of potassium from the extracellular fluid into the cells till the point where we can get to the kidneys and get rid of the extra potassium. Now, if you remember, insulin decreases extracellular fluid levels of potassium. The opposite of having insulin would be not having any insulin or being insulin insensitive, which would be if you're diabetic. If you have diabetes mellitus, you have a predisposition of having hyperkalemia. Because of not having the insulin, you're not going to be able to move that potassium from outside to inside the cell post-meal. If you've got Addison's disease, remember with Addison's disease, the adrenal cortex is not producing the various steroid hormones that it produces, such as aldosterone. So if you don't have sufficient aldosterone, after you've consumed a meal, you won't be able to shift that potassium into the cell, and therefore could lead to hyperkalemia. Now, other things to kind of keep in mind are the effects of the beta-adrenergic receptor. If you have a compound that's an agonist to the beta-adrenergic receptor, that increases the uptake of potassium by the cells. But people on beta blockers, they would not be able to do that. And so people on beta blockers, it creates a tendency for hypokalemia. Now, if you look down here for strenuous exercise and cell lysis, they kind of go, they're going to be very similar, hand in hand. Cell lysis, if the cell membrane gets weak and the cell opens up and lyses, all that potassium that's inside the cell is going to leak out into the extracellular fluid. So, okay, that makes sense. Cell lysis could lead to an increase in extracellular fluid levels of potassium, which can lead to hyperkalemia. If you have a serious enough crushing injury, damage to cells, this is one of the things that can cause some serious consequences for a patient with a major injury, which hyperkalemia can result from it. Also, if you have extreme strenuous exercise, you can damage the cell again, which would lead to potassium leaking out of the cell and result in hyperkalemia. So if you want to kind of put this exercise with the beta blocker and the diabetes mellitus together, I'll give you kind of the perfect storm example. You need to watch out for people on beta blockers or people who are diabetics that are severely exercising. Their chances of developing hyperkalemia are way elevated because of what we just discussed here. So people on beta blockers and diabetics got to be very careful if they exercise too strenuously because they had a predisposition for hyperkalemia. Now, don't worry about alkalosis and acidosis. I'm not going to ask you about those. But I want to mention this one, the increased extracellular fluid osmolarity, just because it's a good time to kind of review what determines the movement of fluid into or out of the cells. So here's the scenario. If my extracellular fluid, the osmolarity of it increases, which could happen with diabetes, because if their blood glucose levels go up, osmolarity can increase. So if the osmolarity increases, where does water want to move? Well, it's going to go from, it's going to move out of the cell into the extracellular fluid because it's drawn to where all those osmoles, the effective osmoles are, in this case, either glucose or something else. So as water moves out of the cell, the intercellular concentration of potassium increases. It's becoming more concentrated. So This increases the gradient for potassium, which will increase the movement of potassium from inside to outside of the cell. Remember, the cells are a little bit leakier to potassium, so if you increase that gradient, you're going to have a little bit more potassium moving out of the cell and can increase or result in hyperkalemia. Now, the kidneys are going to play the biggest role in regulating potassium balance overall, long term. So you have a picture of the nephron tubule plus the ducts. Down here we've got the medullary ducts and stuff, and up here would be the cortical collecting duct. Where I have arrows, if you notice, it goes from in to out. That is representing reabsorption. Anytime you see an arrow that's going into the tubule, that's representing secretion. Anytime you see me put hatch marks on an arrow, that means it's subject to regulation. So if you look at this picture, you'll see the various parts of the nephron in which potassium is either reabsorbed or secreted. Potassium is freely filtered, it's reabsorbed, it's also secreted, depending on where we are in the nephron tubule. Most of the potassium is reabsorbed in the proximal tubule. Almost everything I talk to you about in regards to reabsorption of a substance, majority of that substance will be reabsorbed in the proximal tubule, with the one exception of magnesium. So if I ask you, where's the majority of this particular substance reabsorbed? If you say proximal tubule, you're more than likely going to be correct, because there's only the one exception, and that's magnesium. So majority of it, about 65%, is going to be reabsorbed in the proximal tubule, and we're going to go over the mechanisms of that. You're going to see about 27%, don't worry about remembering the numbers, 27% of what's filtered will be reabsorbed in the proximal tubule, the thick ascending limb of the loop of Henle. And then we're going to look at what happens over here. Depending on the situation, we may have some reabsorption of potassium, but you've got to have a very, very low potassium intake for this to happen. But so this is kind of like an extreme situation. But in the late distal tubule and the cortical collecting tubules, we'll have secretion, and it's subject to regulation by aldosterone. So let's look at the mechanisms by which we are going to reabsorb, say, in the proximal tubule. Mainly in the proximal tubule, it's going to go through the paracellular route, and it's going to be passive from high concentration to low concentration. And paracellular route means we're going in between the cells, not going across the cell, but in between the gaps in the cells. It's kind of being driven, coming along with everything else. In the thick ascending limb, remember we have that 1 sodium, 2 chloride, 1 potassium co-transporter, which again, this is all driven by the sodium-potassium pump over on this side of the membrane. In the luminal membrane, you have this co-transporter that moves all of these in. So in the thick ascending limb, potassium is reabsorbed actively. Once you get later on into the tubule, when you get into the latter parts of the tubule, everything depends upon aldosterone or depending upon how much potassium that you've been taking in. So I'm going to fill in, you have this picture in your notes, and then you can fill in some other things a little bit later. This is the scenario that you would see in the principal cells, which are found in the late distal tubule and the cortical collecting tubule. The potassium is going to be secreted here, and it's pretty much under the regulation of aldosterone. Now, aldosterone will stimulate the activity of the sodium-potassium pump, which allows us to reabsorb sodium but secrete potassium. The aldosterone also increases the number of potassium channels on the luminal membrane, so it makes it easier to secrete the potassium. So we have specific or special channels for potassium that are permeable to potassium that are regulated by aldosterone. This is the predominant means by which we secrete potassium in the late, and I have to emphasize again, the late distal tubule and the cortical collecting tubule, and it depends upon the levels of aldosterone, so it's going to be variable. Now, if you've got a very low potassium intake, you're depleting yourself of potassium, you will not secrete potassium at all in the principal cells. Instead, the intercalated cells are going to kick in. And in the intercalated cells, you actually will get potassium reabsorption. What's going to drive this is you have a pump on the luminal membrane, which secretes hydrogen ions in exchange for potassium. And over on the basal membrane, potassium, you have potassium channels that then the potassium is just going to go across the cell, then eventually into the interstitial fluid and then into the blood. But you got to have, this will only apply with conditions of very low potassium intake. So in your notes where it says distal tubule and collecting tubules, and it lists high potassium intake, low potassium intake, under high potassium intake, you're going to say there's an increased secretion by way of the sodium-potassium pump in the principal cells. When you've got low potassium intake, you've got the intercalated cells, and you're going to get absorption by way of the hydrogen-potassium ATPase in these cells. So it's going to depend upon the level of potassium that you've taken in. The primary regulation of potassium excretion. So remember, potassium excretions mean if it's excreted, it eventually makes its way out of the body. Primary control is definitely going to be by those principal cells. So you saw those principal cells, and you're going to be looking at either affecting that sodium-potassium pump. You can do it by changing the gradient. If you got a lot more gradient for potassium secretion, you're going to favor excretion of potassium. But you also can change the permeability of that membrane for potassium. Again, you're going to see the link to aldosterone. So, few things, primary, what I want you, before we move on, to remember for potassium excretion. There's two primary things that determine potassium excretion. Aldosterone is one, and the levels of potassium in the plasma. So, let's just write it down. More times you write it down, more likely you're going to remember. The higher the aldosterone levels, or the higher the potassium levels in the plasma, the more you secrete potassium, and therefore the more you excrete it. Okay? So that's what you need to remember. So let's look at, these are kind of pretty much what I'm talking about. I'm going to probably ignore this one, so do not worry about this. So we're going to look at these two, aldosterone and the amount of potassium in the extracellular fluid, which favors secretion. We'll mention just briefly about acidosis and it decreasing secretion, but just briefly. So if you look at this, you can fill in your little picture in your notes. You'll notice that if I have a high potassium intake, it's like, I got a little excess potassium, I don't need it, I want to excrete more of it. We talked about short term how we can control it until we can get to the levels of the kidneys and get rid of it. We talked about aldosterone and we talked about insulin. But here we're now down to the kidneys. If I've got higher potassium intake, the levels of potassium in my blood are going to be higher. What that will do is it will directly increase the sodium-potassium pump, as well as increasing aldosterone, which again increases sodium-potassium pump. Both of these will increase the gradient, the potassium gradient. So if you increase the sodium-potassium pump, you're going potassium from the interstitial fluid into the cell. That makes a favorable gradient to get it out of the cell, cross the luminal membrane, and excreted into the tubule. So you're going to increase the secretion of potassium in the tubules, and you could put not just cortical collecting tubules, you can also put in here late distal tubule and the cortical collecting tubules, which ultimately you're going to have an increased potassium excretion in the urine. Now this picture shows you the relationship between the levels of potassium in the extracellular fluid and the amount of potassium that appears in the urine. So you'll notice as the levels increase in the blood, the levels of potassium that's excreted goes up. You don't need it. You got extra potassium, just get rid of it in the urine. Now these graphs are again from Guyton, which shows you the relationship between serum potassium concentration and the plasma aldosterone concentration. Potassium is a potent stimulator of aldosterone. We covered that when we did endocrinology. So higher potassium, higher amounts of aldosterone, because think again what aldosterone does. It favors the secretion of potassium. So you notice over here, as the aldosterone levels go up here, the amount of potassium excreted in the urine increases, and it's pretty linear. So biggest things, if you've got increased potassium, you're going to excrete more potassium directly and also by way of aldosterone. Now aldosterone is definitely going to favor the secretion of potassium through a couple different mechanisms. Remember, aldosterone is a steroid hormone, which means it can easily pass through the cell membrane because it's hydrophobic. It binds to a receptor inside the cell, eventually makes its way to the DNA, and it acts as a transcription factor. And one of the things that increases the production of is the sodium-potassium pump. So increases the activity of the sodium-potassium pump, which makes that favorable gradient for potassium to be secreted. Plus also on the luminal membrane, it increases the amount of potassium channels, which you see here. It increases the potassium channels, so then you can get rid of it. Remember, it also does increase the amount of sodium because aldosterone also favors sodium reabsorption. But since we're talking about potassium regulation, we didn't go that far into the sodium. But aldosterone increases the production of potassium channels in the luminal membrane, plus increases the activity of the sodium-potassium pump, plus also actually produces more of that pump, so you can secrete more potassium. Now this is a picture that's in your notes that just kind of goes over again the role of aldosterone, feedback mechanism by which aldosterone plays a role in potassium maintenance. So I'm not going to cover this. You can look at it in your notes. Now this one's a little tricky. I filled in the picture for you. I'm just going to tell you kind of the overall big picture you want to get from it. In certain conditions in which, such as like if you're consuming a lot of sodium, if you've got volume expansion, a lot of fluid volume expansion, these conditions can increase flow rate in the tubule. And what we want to do is, despite this increased distal tubular rate, we want to be able to excrete the right amounts of potassium and not have too much or too little, pretty much keep potassium excretion the same despite these conditions in which I'm consuming too much sodium or I've got volume expansion. There's certain things that kick in that's ultimately going to cause potassium excretion to be unchanged. You're going to get the amount of potassium you need out. So let's see if we can discuss it a little bit, and hopefully it makes a little bit of sense. So if you're consuming a lot of salt, which is just sodium, what that does, and you can fill this in somewhere, a lot of sodium, because it affects on the macula densa cells, renin levels are going to be decreased, which decreases angiotensin II. Okay? So those two, if you decrease angiotensin II, couple things that will do. It will decrease the amount of sodium that you reabsorb in the proximal tubule, because angiotensin II does play a role in stimulating sodium reabsorption in the proximal tubule. So you have less proximal tubular sodium reabsorption. Also, with that increased sodium intake and the effects on angiotensin II, glomerular filtration rate increases. Overall, both of these are going to cause more fluid to be going through the distal tubule. Okay, so we're down late on into the distal tubule and the nephron. So you increase this fluid delivery, which could ultimately increase more potassium or allow potassium to be secreted. Okay. Well, look over here. Aldosterone levels are going to be decreased if you got a lot of sodium. No need to have the aldosterone level. With decreasing amount of aldosterone, you're going to decrease the amount of potassium being secreted. So if this decreases the amount of potassium secreted, these increase potassium secretion. They kind of counteract each other. And you could I want you to notice it says cortical collecting ducts or tubules, but it also applies to that late distal tubule. So what we've done is we have kind of counteracted each other. So overall potassium excretion is going to remain unchanged because one thing's going to decrease it, one's going to favor the secretion of it. So overall excretion will be unchanged. I can change this whole graph in which you have, if you have something that decreases this distilar tubular flow rate, that would, that anything that does this would actually decrease the secretion of potassium. But this was a scenario where we said, if I increase amount of sodium I'm taking in, a lot of volume, or even treatment with some diuretics, that can increase potassium secretion. But what we do here, by decreasing aldosterone levels, it kind of maintains the potassium levels that are excreted. This is really complicated stuff. I believe it's complicated stuff. So I usually do not ask you questions on your exam about distal tubular flow rate because it's kind of complicated, like knowing all the little steps. I just want you to think about big, like here to here. Despite changes, huge changes in sodium intake or fluid volume expansion, there are things in place to keep potassium excretion relatively unchanged. Okay, so that's what I want you to get from this. Now, under conditions, if you're acidotic, just acutely, very early on, if you're acidotic, being acidotic, those hydrogen ions will inhibit the sodium-potassium pump. So if the sodium-potassium pump is inhibited, that would decrease potassium secretion. So acute acidosis could technically lead to, and I want you to fill this in, would acute acidosis lead to hypokalemia or hyperkalemia? So let's go through the scenario again. If being acidotic inhibits the sodium-potassium pump, what it would lead to potassium secretion. It decreases it. So you're going to keep the potassium in. That ultimately could lead to hyperkalemia. So acid-base disorders can lead to effects on potassium levels. So, but that's acutely. Chronic, or a period of a few days, you can start losing potassium because if it has effects on the sodium and water reabsorption in the proximal tubule, which ultimately can increase the flow rate to the distal tubule and increase secretion. But acutely, it decreases it. Prolonged acidosis, you can start to lose potassium. But again, I don't usually ask a lot of questions about acidosis and its effects on potassium. Okay, it's mainly the effects of potassium and aldosterone on potassium regulation in the kidneys, and is where I predominantly place my emphasis. Now, calcium, we did spend a lot of quality time about the regulation of calcium, so I shouldn't have to go in depth here because I did spend an entire lecture on calcium, and then I even talked about it a little bit later when we talked about GI Fizz. So you've seen all this about where we have most of our calcium, big thing again to remember is the very, very small percent of our calcium is what is regulated. It's this very small amount. It's that free ionized portion is the one that's hormonally regulated and can cause the problems of hypocalcemia or hypercalcemia. Okay, just review the different effects of hypercalcemia and hypocalcemia. See if you remember which of those two would lead to a Trousseau sign of latent tetany. So remember we showed you that? So which of the two? Is it hypocalcemia or hypercalcemia? I'm not going to answer it. You need to be able to figure that out. Now, how we regulate it? We spent good quality time talking about parathyroid hormone. So what is the role of parathyroid hormone in calcium balance? And specifically now we're going to emphasize its role in the kidney. So remember, though, I could still ask you questions on its effects on bone in the intestine, though, too. Remember, if calcium levels in the blood dip down, and specifically the free ionized levels of calcium decrease in the blood, it stimulates the production of parathyroid hormone. So parathyroid hormone, what's its effects on bone? Does it stimulate bone accretion or bone resorption? Ultimately, what do you want parathyroid hormone to do? To increase the calcium levels back up into the blood. So are you going to build up bone or break it down? You're going to break it down. So this stimulates bone resorption. The osteoclast activity is going to increase. Its effects on the kidneys. Parathyroid hormone stimulates the 1-alpha hydroxylase in the kidneys. How many times have we had that on an exam? Probably more than once. In the kidney, parathyroid hormone stimulates that 1-alpha hydroxylase, which converts 25-hydroxy cocalciferol to calcitriol. So the active form of vitamin D, calcitriol, is formed in the kidney. Calcitriol has effects on the intestine, which increases the reabsorption of calcium, and also the calcitriol also increases phosphate in the intestine, but I'm going to be concerned about parathyroid hormone. So I want to know the effects of parathyroid hormone in the kidneys. So what's its role? Calcium in the kidneys and how it controls excretion. Well, calcium is filtered as long as the free ionized calcium. If it's bound to a plasma protein or bound to like phosphate, any of the anions, it's not filtered. But of the one that's filtered, we can reabsorb it. Now, calcium is not secreted. Okay, so we either, if I want to get rid of some extra calcium, I just don't reabsorb it. If I really need to keep all my calcium, I'm going to reabsorb as much as I possibly can. So keep this in mind. Write it down 2 more times if you need to. Pretty much the different ions that we talk about are reabsorbed, and only a very couple are actually secreted. And the only 2 things that are secreted is potassium, which we just went over, and the only other one, which we'll talk about at a later lecture, is hydrogen ions. Okay, so calcium is filtered and reabsorbed. That's it. It's not secreted. So we look at your nephron tubule. You'll notice again we got these arrows. This represents the reabsorption of calcium in the proximal tubule. Again, majority of the calcium that is filtered is reabsorbed in the proximal tubule. Then the next highest amount that's reabsorbed is in the thick ascending limb of the loop of Henle. We're going to show you the mechanisms of how all this happens. Then you'll notice I got a tiny little bit over here that's in that late distal tubule. And also it actually would apply, we'll just say late distal tubule now. But you'll notice that these ones are the hatched ones. The reabsorption of calcium in these locations are going to be regulated by parathyroid hormone. Okay, so let's go over the mechanisms by which calcium is reabsorbed and the role of parathyroid hormone. So in the proximal tubules, calcium is reabsorbed through the paracellular pathway, which you see here. Most of it's this way, kind of comes along with the water. We do have some through the transcellular pathway, and it is by active transport. So we have a calcium pump on the basal membrane, which drives the calcium that's over here in the lumen to be reabsorbed. So it creates this favorable gradient. We do also have a sodium-calcium exchanger on the basal membrane. Now, something that you might want to take note of is the calcium reabsorption of proximal tubule very much closely parallels the reabsorption of sodium in water, because most of that calcium is coming into this paracellular pathway, which is coming along with water. So anything that influences sodium reabsorption will definitely influence water reabsorption, which could affect calcium reabsorption. So things that change the extracellular fluid volumes or can change blood pressure can affect the amount of calcium that's reabsorbed and then ultimately how much is actually excreted. And we're going to go through a table and show you that. In the loop of Henle, and specifically the thick ascending limb of the loop of Henle, and in the distal tubule, you have both of them has a transcellular route, which you'll notice here I have it starred. That means they're regulated by parathyroid hormone. In the thick ascending limb, you do also have some that goes to the paracellular route. But pretty much what I'm going to ask you, is where in the nephron is calcium reabsorption regulated by parathyroid hormone? And you say the thick ascending limb of the loop of Henle and the distal tubule. And it favors the reabsorption by really biggest thing is affecting the calcium pump. Okay, so the transcellular mechanisms are very similar to what you saw in the previous slide. So you have that calcium pump that drives the movement of calcium into the cell. Now, various things that can regulate calcium reabsorption obviously is parathyroid hormone and calcitriol, and we're going to look at some other stuff too. So I have filled in this table for you, but I want to make sure again you know what these titles mean. So what we have here is if I have an increase in parathyroid hormone, what does that do? It decreases the amount of calcium that's excreted. You can change that if you want to, instead of saying decrease calcium excretion, you can say favor calcium reabsorption. You could, because that's what parathyroid hormone does. But I think these are a little bit better because some of these are not necessarily effects on reabsorption, but they can affect the amount of calcium that's excreted. So these things here ultimately will cause less calcium to be in your urine. These things over here can cause you to have more calcium in your urine. So parathyroid hormone and calcitriol should be easy ones because we've talked about those previously. Increased parathyroid hormone, you favor the reabsorption of calcium in the kidney, so you have less calcium that's excreted. Calcitriol also does the same thing. It decreases the amount of calcium that's excreted, both of those. So remember, when you have a lot of parathyroid hormone around, calcitriol, their job is to try to get blood calcium back up. Now, if you look at the plasma phosphate, the increase in plasma phosphate, what it does is it directly stimulates parathyroid hormone. So if you've got increased plasma phosphate, that increases the amount of parathyroid hormone, because what parathyroid hormone does, besides increased calcium reabsorption in the kidneys, it favors the excretion of plasma phosphate or phosphate levels. So that's something I've asked you more than one time, is what does parathyroid hormone do to phosphate, and what does it do to calcium? It increases calcium, it decreases phosphate because it wants to get rid of it, because phosphate will couple along with calcium, and you're not going to have free calcium around. It's going to be sitting there with each other and not affecting. You're not going to be able to do what you need it to do. Also think about parathyroid hormone stimulates bone resorption, breaking away with the bone. Calcium is going to go up, phosphate's going to go up. It's get rid of the phosphate. So that's what I want you to remember. Now you'll notice if I'm going through this, you just remember the opposite will apply. So if I have decreased parathyroid hormone levels, I'm definitely going to increase the amount of calcium that's excreted. Okay, if I decrease the amount of plasma phosphate, I'm going to increase again the calcium excreted. Now these two kind of go hand in hand. Remember I had mentioned that calcium reabsorption very closely mimics or parallels sodium and water reabsorption. So I said changes in extracellular fluid volume or blood pressure can affect calcium excretion. So here's the scenario. My extracellular fluid levels, they decrease. Okay? That could drop my blood pressure. Don't like that. Well, what's going to happen? Well, if my extracellular fluid volume decreases, my blood pressure decreases, we're going to set a series of things in motion to try to get that extracellular fluid levels back up and the blood pressure back up. And we covered the role of the renin-angiotensin-aldosterone system. So if you have a drop in extracellular fluid volume and blood pressure, renin levels will go up, angiotensin II levels will increase, and angiotensin II will stimulate the reabsorption of sodium and water in the proximal tubule. Plus remember the effects of aldosterone will increase sodium and water, but that's mainly in the late distal tubule and the collecting tubule. But if you notice, if you increase sodium, you increase water in that proximal tubule, calcium kind of comes along for the ride. So that favors the reabsorption of calcium in that proximal tubule. So they kind of go hand in hand with sodium and water. So if you notice over here, if these things are elevated, what that does is it results in decreased renin, decreased angiotensin II, decreased aldosterone levels. You're going to have less sodium and water reabsorbed. And again, they kind of go along with calcium reabsorption in that proximal tubule. Now again, we had mentioned about acidosis, alkalosis. I will not ask you about the role of acidosis or alkalosis and the excretion of calcium, so I think it's too much, but I could ask you any of the other ones. Definitely parathyroid hormone, plasma phosphate, and calcitrol, since we have talked about them in the past. Now the last two things we're going to look at is phosphate and magnesium. Phosphate is relatively easy to remember. Phosphate is either completely reabsorbed in the proximal tubule or it just stays in the urine. That's it. So mainly phosphate is reabsorbed with sodium. There is a co-transporter in the proximal tubule that sodium and phosphate come together, and it's found in the luminal membrane. The way it usually works out, it all comes down to thresholds, or transport maxes. We have a certain amount of transporters that allow us to reabsorb phosphate. Generally, phosphate levels are below that transport mechanism, so it pretty much is completely reabsorbed. But if the phosphate levels exceed that transport max, then excess will be excreted. So most of us, though, excrete phosphate in our urine because we're just going to get rid of any excess that we don't have. So that's primarily called an overflow mechanism. Even though we did mention that parathyroid hormone will decrease phosphate reabsorption, but overall, though, it's by this overflow mechanism primarily. Okay. The last one, of course, I just mentioned that, is magnesium. Magnesium, like calcium, is found mostly in bone. Again, a lot of it's found in bone. What I want you to remember about magnesium, this is like the one weird thing that it's unlike everything else, is most of magnesium is reabsorbed in the thick ascending limb of the loop of Henle. Unlike everything else, most of it's reabsorbed in that proximal tubule, but not this one. It's reabsorbed in the proximal tubule, but the majority, about 65% of it, is reabsorbed here. Then we got a little bit that's reabsorbed later on. Okay? The actual means by which we regulate magnesium is quite not really known, so I'm not going to ask you, because they really don't know it. I'm not going to ask you for it. So the main thing I want you to remember about magnesium reabsorption is that predominantly reabsorbed in the thick ascending limb of the loop of Henle. Okay? Now, one thing that I want to see if you kind of notice a trend is we really don't have things reabsorbed solute-wise, magnesium, calcium, any of that, in the descending limb, because that's mainly just permeable to water, nothing else. You don't see a heck of a lot in the thin ascending limb either. It's other places where you see things like magnesium or calcium or potassium or any of the movement of other stuff. This is mainly would be like water on the descending limb. Here, the thin ascending limb is permeable to salts, but it's primarily going to be a passive reabsorption, and it also is permeable to urea. Okay, so a lot of this other stuff you can see in other parts of the nephron tubule.